So perhaps I think we'll start, because all the talks are still fresh in people's memories, perhaps we'll talk with, we'll start with any specific questions that members of the audience might have for the speakers. And again, I would encourage students to, please don't be afraid to ask questions. It's a fantastic opportunity when you have experts like my colleagues here in the audience and are here, and you get to ask them questions directly. So please ask questions.
Aceitamos questões em português, inglês e espanhol. Ótimo. Marília, o botão tem que apertar e segurar para ele ligar. O LED verde, né? Ontem eu testei.
A question for Mark. It's very interesting the analysis you do in cortex in with epileptic activity. The focus, as far as I understood, is in the cortex. However, it is very well known that surrounding the focus area, there is an area with inhibitory mechanisms in order to avoid the spreading of the epileptic activity. Have you ever analyzed that area?
So that's a very good question. So obviously there is this granularity which we observe with the interictal activity. If you remember in the early slides I showed you this ictal event, and some of the work that we're now trying to do is look at the propagation of activity from this region, this hotspot region, where we obviously see these anatomical changes, to these surrounding regions during the ictogenic process.
Hi, thank you for their presentation. And my question is, considering the sickness behavior, what do you think, Professor Gabriel, about the treatment of depression associated with epilepsy with anti-inflammatory drugs and anticonvulsive drugs, for example, an anti-inflammatory drug and valproic acid?
Thanks for the question. Yeah, in this review, they go along saying that depression, like sickness behavior exists until you have the pathogen in your body. So after the immune system is successful in getting rid of the disease, the behavior stops, right? So the difference is that in depression, you don't have anymore an external agent, and the brain is like caught in this state of depression. So the thing is that the immune system is the trigger to this, so you need the trigger, maybe you don't need. You have genetic alterations that the more people, like depression also has a strong family tie, so runs in families, is a strong genetic background. So is this genetic background related to immune markers? And there are some papers that show that you have also immune differences in depression, family history of depression. So what happens is that's the big difference. So sickness behavior stops when you remove the pathogen, and depression stays. So for the part of the comorbidity that you asked, of course, like since that there was the presentation showing depression as a comorbidity in epilepsy, and of course when I think like, well, if you have these cytokines involved in epileptogenesis and you have this also the role of the immune system in depression, so are they correlated? So it's an epiphenomenon or they are correlated? That I would like to research further. I don't remember if there was any publication showing this already. I don't know. If it exists, I don't know. You have to look. Thanks.
Hi.
Claudio.
Thanks for your nice presentations, all of you. I have a question for you, Mark. I was pretty impressed by the optogenetics used in human slices. As far as I know, if you use optogenetics tools, you need some time for the virus to express the opsin channels. So I was just wondering, first, technically, how do you see, or how can you make the virus increase its production rate in order to make, well, to the slice, is it still alive with the opsins levels high enough so you can stimulate the cell or inhibit it? And the second is, in a way, future directions. From your point of view, do you see optogenetics as a real treatment for, let's say, not in vivo neocortex, but in vivo? So do you believe in the future we will be able to inject a virus with opsins in the healthy brain or unhealthy brain to treat disease? What's your opinion about it? Thank you.
Great. Yeah, it's a great question. Well, we at Newcastle, we've just obtained £13 million from the Wellcome Trust and the EPSRC to do exactly that. So by the end of 7 years, we're going to be putting an optogenetic tool, a closed loop system, into an identified human patient. Now, obviously, that's going to involve a huge degree of work, which will involve not only the type of basic science that we can do at the level of a human slice to identify what cells we need to specifically target. It also will involve the development of a non-human primate model of epilepsy, because we need to be able to test it in a primate model. And in addition, developing the optogenetic tools that will allow, as you've had already alluded to, safety in terms of the introduction of a virus into the human brain. It's clear from other fields of biomedical research that gene therapy is now being used in other organs in the human body. Just recently, for example, in the United Kingdom, a trial has started using gene therapy, viral targeted gene therapy in cardiac patients. I think there's probably a degree of Frankensteinian fear associated with actually introducing these into the human brain. But clearly the funders think that we can do it, so hopefully we can. Just coming back to your first question about the opsin expression, I mean, I should point out that the data I showed today is simply, all I can say is that we've had successful infection with GFP and using a CAM kinase specific promoter. Obviously, the next step is to do it with the opsins, and we do envisage that probably we're going to have to play around in terms of the time period that we need to use to get good viral opsin expression. Clearly, we were very encouraged with the short time course that we need to get the virus in there. I mean, HSV is a fairly potent virus, and again, one that you wouldn't want to be using in a human. Probably you'd be looking more at a lenti or an AAV type virus. But clearly, we're going to have a lot of work to take it to the next step, which is to try and work out what is the best time scale. As I say, we have optimized our approach in that we can probably keep our tissue alive for up to about 24 hours. Now, that may not be enough time to get adequate viral expression, but one of the things that we're also developing, which I didn't talk about today, is we're culturing our human tissue. And again, if we can get successful cultures, that again means that we prolong the window in which we can do this type of work and do the types of recordings that we need to.
Good morning. Well, first of all, I'd like to thank you all for the presentations. You have no idea how much I'm learning in this Congress. Well, I have two questions. Firstly, to Professor Gabriel-Arisi. It's amazing the role of the immune system and the interleukins and TNF in the central nervous system. And I'd like to ask you a question about what do you think of maybe we use some anti-inflammatory drugs, steroidal and non-steroidal drugs, as pharmacological tools for research and also for treatment of epileptic patients. And secondly, for Professor Lydia. It's interesting that the hippocampal neurons in CA1 area die, many of them. And I'd like to ask if a researcher has an idea about which types of neurons die. For example, symmetric synapses, asymmetric synapses, or for example, in questions of neurotransmitters or something like that. And thank you.
Yeah, I don't, and the last part of the question that you did, I don't understand. What do you mean? About the neurons or about the synaptic contacts of the neurons?
Yeah. You have said in the presentation that a huge part of, say, one area neurons die.
Yeah.
Which kind of them? For example, GABAergic, inhibitory neurons, glutamatergic, excitatory neurons, or something like that, if someone has already researched about it.
Okay, I understand. In my studies, what I know now is that there are no neurons. So we really don't know if the remaining neurons are only pyramidal neurons or interneurons. But in the CA1 region, I have also studied the staining for some interneuron markers, parvalbumin, and there is a dramatic lack of these interneurons. So I think that the few surviving neurons are pyramidal cells, mostly. I can't say that all of them, but mostly. If it's enough for you, or do you need more information? Okay.
So answering, I think you asked about the drugs and the biopharmaceuticals, right? So, I don't know if it's on, yeah. So, to deliver the biopharmaceuticals to the central nervous system, probably you're going to need a vector. So, these will be the next steps. I don't know. Of course, you're going to need a huge effort to work with that. I don't know if the industry or the academia will work it out. And about another drugs, there was a very promising drug. It's called minocycline. Minocycline is an antibiotic and has anti-inflammatory properties, and was tested in a phase 2 trial, I believe, in the US. I don't remember for what. If it was ischemic damage. Do you remember this one?
I know that it's been touted as for schizophrenia and psychosis.
Schizophrenia, yeah. But I remember that the phase 2 trial, I think, was a huge one. I think was ischemic damage, and the problem was that the patients have a lot of side effects, and they have to stop the trial. And then, because the results in the experimental animals were great with minocycline. I tried also minocycline in the pilocarpine model, and the results were good, were promising. The problem that they realized, there was a published paper later on that shown that the dose-dependent cure for minocycline is like a U shape. So if you go for a high dose, you have a lot of side effects, and it was very problematic. And the dose they tried in the phase 2 trial was really high for the patients. So it was a daily doses. I don't remember how many milligrams, but was too much. So they should try again with lower doses. And minocycline is very promising. It looks like very, very promising on different things, actually. Also, like you said, in schizophrenia and maybe in epilepsy too.
Okay, but I just wanted to add there is a caspase inhibitor that is being tested now, or has been tested for treatment of epilepsy.
I'm sorry, I could not hear you. Is the microphone on? Because from
the,
here we can listen. It is? Okay. Okay.
There is a caspase inhibitor that is being evaluated now for the treatment of epilepsy in the United States. So that may be one possible drug that has no anti-epileptic targets. It's basically targeting inflammatory processes. Right. But it's being tested as an anti-epileptic drug, so we'll see what that shows. But I wanted to ask you, Gabriel, a more methodological question. If you were to study, the impact of the interleukin and the different interleukin in the clinical domain with patients with epilepsy, given the variability in the concentrations. First of all, where would it be best to get the samples? In CSF or in serum? And when would you get those samples? Let's say if you do it in the serum, would you get samples postdictally, interictally, both? And what would you expect to find based on what we know so far?
Well, yeah, there were some studies, like the study that I cited, Sinha was made in Bangalore. He was made with CSF. There was another one, Peltola, I believe, was made after seizures. So they collect CSF because they have the spinal tap for another reason, and they collect CSF and they measure cytokines after seizures, and then they got IL-6. So to identify for practical reasons, like here in the surgical center in Ribeirão, you can do the same kind of study that they have performed in Netherlands because they have the tissue. So you can go for the tissue, for the macerate of the tissue. But for me, nowadays I'm thinking like we lose the structure. So it doesn't make sense to measure the cytokines without the structure of the brain because the information is in the structure. So you have to keep the structure and then go after the cytokines and also the receptors. And then you can jump to a functional study of in vivo like Cunningham is doing with cultures of the cells taken from the patients and then check about the inflammatory process. The problem is that the patients that are reaching surgical service, they are far away, like many years into the disease. So you don't have you have the post hoc information. You don't have the information from the beginning. So that's why CSF taps will be great in post-traumatic brain injury or fever, seizures in children, when you can measure these cytokines and realize which ones are the triggers right after the precipitating injury. So this will be the studies that we need to be done. And I don't know if someone is going after that. I hope someone is. Is the answer to your question?
Yeah. Congratulations, Mark, Gabriel, and Lydia for wonderful talks. I have a question and probably a comment regarding what Gabriel said about the sickness behavior and what would be the overlapping with depression. And I'll comment one thing that happened with me, but I have a colleague, he's a psychiatrist, and last year he said when he had an infection, for us Brazilians, many of probably in the audience, we probably had dengue, and he commented with me that, João, this is, of course, he stayed a couple of days really with a kind of flu, but the depressive behavior or the sickness behavior lasts many weeks. And he mentioned to me that, oh, this is the closest thing that I can identify myself with depression, with my patients, depressive patients. And unfortunately, this year, I got dengue. And I think that the feeling of sickness, although the disease is well, we know very well that in the acute inflammatory phase, we have like a breakdown of the vascular permeability. So the hypotension is very important to the sickness behavior. But after a week, you were fine, you stopped drinking water, but the feeling of depression stays for at least 1 or 2 weeks. It's sometimes even longer. So I think that maybe, I don't know if people in the audience who had dengue also had the same feeling, but this is, and I remember what my colleague had commented to me and said, oh, probably this is what I think must be depression. And probably if you are commenting that this feeling of sickness usually goes away when the disease is over, probably looking at some of the interleukins after or during and after the dengue infection would be very interesting. We know that probably the interleukins would last longer in dengue because the permeability would actually leave part of these molecules in the interstitial space. But I think it would be a very interesting, I don't know if you, Gabriel, knows if someone has a look at dengue, because probably, at least for us Brazilians and for some regions in Brazil, it happens, and this is a comment that probably is not my only experience and my friends.
Yeah, just to say to you, it's like, well, I checked it yesterday at PubMed, and cytokines and nervous system, they are being published 2,000 papers per year. So, yeah, 2,000 per year is how long? How many per day? It's like 6 per day. So, it's heavy to keep up. But for sure, João, they are making good works about this, about the immune scare of the signature that stays in the body and predispose you to another one and another one. So people that have recurrent diseases and they are prone to have disease because of this sort of things is very hot field of research right now.
Over here, I have 2 questions for Lydia. Nice presentation. The first question is about devascularization. Since there is a reduce in vascularization in hippocampus and cortex, do you think there may be a possible explication for this is that considering reduced metabolism promotes neuroprotection, if it's the case? And the second, if refractoriness is patients that don't respond to drugs are related to reduced vascularization and reduced glycoprotein B?
Okay, I'm going to answer the first part because I can't hear properly the second part. And about the reduced metabolism, I think my studies are only structural, so I don't have the functional information. So what we think now is that in this region in which the blood vessels are not working properly, I think there is a reduction in the metabolism because there are almost no neurons there. So in this region, nothing is working properly. So this part of the hippocampus is like dead. It's like it's not working, and maybe we think that in the hippocampal sclerosis, this part of the hippocampus is not responsible for the hyperexcitability. And now we are looking where is this hyperexcitability starting or recruiting the activity to make the epileptiform activity.
Glia also gliosis is very present. So do you think there is not a huge demand for glia sustentation since there is a huge gliosis?
I only catch a part of your question, and I suppose that you're talking about the gliosis in this area. I couldn't hear.
It's hard to hear from here. Do you want to ask in Portuguese or no?
No, but I think we can hear because of the echo.
Yeah,
it's
the echo,
it's horrible here. We
to
can't hear almost anything. It's about the...
If you said that since the tissue is dead, there's no need for vascularization, but there is a huge gliosis.
Yes, there is. Yeah, I understand this part. So you think that if the part of the hippocampus is full of glial cells, but no neurons, is not there. But the glial cells are not spiking cells in theory. And so under the point of view of the epileptiform activity, I think that this part is not responsible for the activity, but the reduction in the blood vessels in this part, considering the gliosis, is difficult to understand because these glial cells have to survive. But the glial cells are hard. I think that you can find glial cells in any kind of disturbance in the brain, and almost they are like a survival cells, and they don't need as the same blood supply that the neurons, and maybe they survive with the very few blood vessels that remain, because there are some functional blood vessels in this region. But anyway, in this kind of study, we have also another problem when we consider that the functional imaging techniques, like the tomography, or I don't know the name, maybe the functional techniques of imaging, they report changes in the metabolism. And I don't understand how the blood supply is changing if there are no useful blood vessels in this area. So we think that we have a lot of work to do in this region to understand what is going on.
And secondly, the second part is about the refractorities and blood vessels, since there is a reduced drug delivery in the system.
Yeah, for sure, because the drug resistance of the patients are related to the disturbance in the blood vessels. There are several studies regarding the diffusion of the drugs, like a problem in the target of the drugs. And maybe this is a possible explanation also related to the resistance of the treatment of these patients. But in my case, I mean, in these, the patients that I presented here, all the sclerotic hippocampus are coming from patients that become seizure-free after the surgery, because we prefer to study only these patients because we consider that the epileptic region has been removed, because the patients are completely seizure-free after the surgery. So,
I mean, just following on that point about gliosis, I mean, we see huge gliosis in the neocortex as well, and I agree with Lydia in that they're not spiking in that, you know.
You must know that I don't know.
in the conventional sense, but you know, ultimately epilepsy arises from networks of cells, and the glial cells have a massive contribution, for example, in extracellular buffering of potassium, which again we know plays a role in epileptogenesis. So I think really, you know, we need to think about all of these things. It's clearly multifactorial, it's very complex, and this is kind of something that I'm going to, if we have enough time, talk about in the discussion bit of this. So
can I make just a short comment?
What?
Because I feel between Cajal and Golgi here. Like 100 years later, Cajal is right by my right here, and Golgi is vindicated by my left. So
the syncytium.
Yeah, the syncytium. So okay, you were like, Leah, don't spike, and okay, and he was like almost jumping over me.
Concerning that issue, don't you think that the overexpression of the glycoprotein that avoids is a transport that avoids the introduction of drugs and epileptic drugs into the brain? It is very well known that that protein is overexpressed, but also it could be expressed in neurons and glia. It is possible that the few neurons that remain in the epileptic focus overexpress the gly protein. But in your case, have you ever seen any expression of this protein in the vascular changes you observe? Because you observe a lot of changes in these vessels. I suppose that they have an overexpression of this protein.
I didn't do these studies, but I think that I have to do. I mean, the P glycoprotein that I read about that, it's critical in this epileptogenicity of the tissue, I know. And I didn't know that it was expressed also in glial cells, as you said. So, and the protein is related to the drug resistance. So,
because it could play an excitatory role in neurons.
In neurons?
Yes.
It's like in the survival, you can consider that the survival neurons are overexpressing this protein, so they can be like hyper-excitable neurons or something like that?
That's right. That is the hypothesis.
It could be nice to find an overexpressive there. I have to try.
And the second question, is there any relationship between the changes you observe in the vascular changes and the duration of epilepsy? For example, a patient with longer duration of epilepsy present more significant changes?
I didn't study. I have to try. Thank you for your nice ideas, because I have to perform new studies. And I don't have any idea about that, but I have the data. I have to review my data about that.
Here? Oh, Norberto first. Yeah.
Yes, thanks for a marvelous discussion. I didn't go back to the question of the triad, the neuron, the endothelium, and the glia, because I think you are kind of sliding from the answer. Because I think even 100 years later, you will have tools today to compute things, as you are showing brightly with ultra-structural dimension. You have conditions to measure at that level combinations of factors, synapses. So what if you combine this with whatever number of inflammatory agents, obviously glia is in that. You're measuring glycine, which is part endothelium plus glia. You have neurons that we can measure at some level in a composite situation. So what if you have the utopic ideal response to this? If you have the tons of pounds or thousands of pounds, and the IBM project, Grupo project has trillions of thousands or whatever money. So what if you have that power today and say, okay, these three and something else is involved? What is the solution? Because inflammation is in your tissue. You didn't talk about that. Yeah. You have neurons in your tissue and they just disappeared. So disappeared means you denervate something, which is hyperexcitable because of denervation. It's a very classic model of hyperexcitation. So what if you have the ability, the three, please, to answer, if you have the trillions of pounds or whatever, to say, okay, this is, just to speculate with responsibility.
Norberto, you always have to go and steal my thunder, don't you? That was exactly the sort of the way that I was going to sort of get. I mean, we've already had questions for half an hour now. I think probably we've very nicely segued into the discussion part, the sort of open discussion. But I mean, I think that's exactly the point that I was going to raise, is that clearly there is this central role for this idea of the sort of tripartite synapse. Again, this was one of the things that was raised in this document that ASLA was a co-author on, this outlining the future work, future directions, where in part of that document they raised the point of understanding neuronal, glial and vascular changes that occur. I think what we have to bear in mind is that there's obviously all of this very detailed data that is emerging from the studies of Gabriel, from Lydia's studies. The problem from a human point of view, at least for myself and Lydia, is that it's always endpoint. You know, you're seeing this tissue at the end. But clearly, I think we probably need to take a sort of very out-of-the-box approach, in that we can take these findings and then sort of back engineer and put them into animal models. And then what that allows you to do is actually look at how each of these elements contribute to the process of epileptogenesis in the brain. So that clearly is a big question, and one that I think should be addressed, but not by my ground. Sorry.
And also, as Luisa suggested, sometimes the data are just in front of me, and I don't know what I have to look for, any other extra details, like compare the duration of the epilepsy and the results of my staining or whatever, because sometimes we have so many data that we don't find any relationship between them, but there is. And maybe this is critical for understanding the epileptic process, or how this brain is getting worse, and the epilepsy is going to be finished after the surgery or not. I think so.
So I have to answer. Can I run? Can I slide? Like you said. Okay, let's try to. When I think the first time I read this expression like triad and synaptic triad was when I was learning how bold worked. And I was looking at the thing and I said, well, but they have this fine control, so we have the fine vascular control, point to point, moment to moment, and every tissue has this. Of course, you have every liver works this way. If you have greater metabolism, you have great vascular supply. But then in the brain, you can use the brain, only the blood supply, to measure brain activity, the electrical activity. And then I was so surprised, I was thinking like, wow, wait, we have to go after that. So we have to treat the brain as an organ, as a regular organ. It works like the liver works, and it works like the kidney works. This was when in the press symposium, I was thinking about, we are so marveled by the neuron structure, and when we are so marveled by the electrical activity that is so invisible and mysterious that maybe we are wondering for maybe 100 years after that, and we forget this, keep it simple. Like, yeah, you have the vascular and you have the blood, and it's an organ and need this. So if you go after that, you can discover what's going on with the electrical activity. And I have questions for both of you. Can I make? Yeah,
yeah, sure.
Yeah, sure. So first for Lydia.
Okay.
You just said that you have all this data, and I was looking at your data with the synapse. So you have always talking about the symmetrical and symmetrical, but I was wondering, have you ever checked it? Because in the excitatory synapses, you normally have an area with AMPA receptors and another one with NMDA receptors. Have you checked for differences in area and shape?
This is future work. I mean,
future work.
I have too many things to do after this meeting. And,
you have a long flight back home, so.
Yeah, I have too many things to think about that. Maybe if we try the immunogold staining, we can localize precisely the receptors. But it's difficult technically, I mean. And as you said, we can try to understand how the excitatory synapses are organized, or maybe if they are altered in the composition of the AMPA or any kind of subunits or receptors.
I mean, there is a new, just to say, you might not have to use immunogold. There's a new high-resolution confocal approach, which is called STORM, which Istvan Katona has been developing in Budapest.
I know.
And the resolution, I mean, is amazing, particularly in terms of trying to resolve at the level of receptors. So
which is the resolution of this microscope?
I mean, I can't remember off the top of my head, but I mean, it
less
is...
than... it's about 0.1 micron.
40 nanometers, yeah, 40 nanometers.
Well, it's perfect.
So, yeah. So, I mean, STORM is emerging as a very powerful new technique that will allow sort of the invasive resolution at the level of synapses.
So, Mark, my question to you is also synapse. So, you said that most of these are electrical coupling.
So our idea
gap junctions
is that there's a role for gap junctions. Yeah.
Okay. So have you checked it when you showed EM?
Yeah.
You talked about symmetrical and asymmetrical synapses, and I was waiting. Where are the connections? So have you ever checked this with immuno or EM?
So looking for gap junctions can be a little bit like looking for a needle in a haystack, particularly with EM.
Yeah.
But one of the things we're doing is we are working with a group in the United States who have some very good antibodies. I mean, that's the other issue with gap junctions, is that the antibodies are not great. And you need to know, is it connexin? Is it connexin 36? Connexin 36 are thought to be the ones that mostly connect up the GABAergic interneurons, which are out on the dendrites. We believe that these gap junctions exist between the axons of principal cells, and it's believed that they may be formed by panexins. So really, it's trying to. The issue at the minute is a technological one.
Yeah, and also you need an excellent conditions of feed section.
Yeah.
And I have a question also for you. It's about the...
Well, you talk amongst yourselves. Okay?
If, yeah. It's about the gap junctions.
Yeah.
I thought that the parvalbumin interneurons have also, they are very frequently presenting gap junctions between them. But in the epileptic tissue, there is an alteration in the parvalbumin positive interneurons, as you said, and as we saw also. And my question is, do you think that the parvalbumin is losing the expression of the protein, or the interneurons are losing are dying?
I knew you were going to ask me that question.
No, I mean, it's because I have my second part is if interneurons, the parvalbumin interneurons, contains the gap junctions, probably, and in the sclerosis, in the epileptic tissue, these interneurons are altered, and maybe these interneurons, which are originally for establishing the gap junctions, they change in a mysterious way, and the very fast oscillations that you observe maybe are linked to these alterations in this interneuron subpopulation. Just to speculate.
No.
I don't know.
It's great. I mean, this is the type of things that we want to throw out there as part of the discussion. So, so let me try and address that. So first of all, we have some preliminary data in which we've to address this question of, are we simply losing parvalbumin protein, or are the interneurons themselves dying? So we've stained for GAD, and we don't see any significant changes, which would suggest that it's the PV signal that's going down. Now, what's the ramification of that? What does that mean? And one of the ideas that we have is that there is this phenotypic change. So parvalbumin is known to be important for calcium buffering. It's also strongly interlinked with NMDA receptor expression on parvalbumin-containing interneurons. And what we believe is maybe occurring is that parvalbumin expression goes down, you lose the protein, and you actually then phenotypically change the behavior of the interneuron. That, in addition, may alter gap junction expression. It's highly likely. We don't have any evidence to suggest that, but that could be a very likely possibility. And then that in itself sculpts how your network oscillation changes itself. So I think there's lots of questions, which is always good,
for sure.
because that's what drives future research. But our feeling is that the interneurons themselves are not dying. They are simply becoming different in terms of their electrophysiological behavior.
Yep.
Hi. Can you hear me?
Yes.
Okay. Thank you for the great talks. I was just wondering, Mark, did you characterize very fast oscillations in the normal brain tissue, and do you see, qualitative difference between these very fast oscillations in the range that you record, that you define, very fast oscillations that you could create, let's say, a data table of regular, very fast oscillations, or these are very particular to the epileptic brain sections that you have, and coupled to that, consider that you have a slice of human brain tissue. How long range can you record? Because what I'm trying to think is, what is the theoretical framework that you're thinking when you're looking at the damaged brain tissue? You're looking at where the sclerosis is, or where the penumbra area, or where the area that still has some neurons. What actually, how do you theorize your research line in terms of how to put this circuit change these functions?
Okay. Great question. So in trying to address the first one, first of all, I would just reiterate, we don't see spontaneous interictal activity in the control tissue. So when you have a normal brain slice from a patient that does not have epilepsy, these are these patients where the tissue's been removed because they have an underlying tumor. it's essentially, you actually get the presence of spontaneous rhythms that are associated with kind of normal function. So you get some beta, you get a little bit of gamma, you get some delta in there as well. And we don't see these very fast oscillations that are clearly associated with these interictal sharp waves, but also occur on their own. In trying to classify these events, which I think was the second part of your question, because I think actually there's 3 parts probably. We've just had a paper accepted in European Journal of Neuroscience, which is, it's pretty computational. There's some human data in there as well from our brain slice recordings. And what we've done there is we've gone back and looked at our data in much more detail and actually tried to break this activity into ripples and fast ripples. So I sort of mentioned it during my talk that there's this debate in the field at the minute whether you're a lumper or a splitter. So some people just lump high frequency oscillations all together and say anything between 80 and 500 hertz, that's the bad oscillation. But other people would suggest that actually you need to split it, and you need to split it into ripples, which are 80 to 200, and then fast ripples, which are 200 to 500. And there is some work that's emerging which suggests that it's these fast ripples which are more strongly implicated in epileptogenesis. And this comes back to the fact that you can see physiological oscillations that are within that ripple band, so 80 to 200. I mean, there's work that's been produced, for example, looking at memory consolidation, and you see ripples there. So I think when we think about this type of oscillatory activity, we have to be very careful, and future work will really try to discern which rhythms you base, say, a clinical decision on to resect a certain part of the cortex. My feeling is that you need to go after these fast ripples, that they are the better candidate in terms of epileptogenicity. And then the final part of your question was this idea, the theoretical idea of how we can, as I think what you're trying to get at is scale up the work that we're doing at the level in vitro back to the in vivo situation. One of the things that we're doing in Newcastle is we are trying to develop a clinical service whereby we're doing subdural recordings, but with much finer spatial arrangements of the subdural grids. Because at the minute, the classical clinical approach is to use large macro electrodes. However, a number of the companies that produce these subdural grids are now producing grids that have smaller electrodes, but also smaller distances between those electrodes. Because, again, this comes back to the paper that I mentioned that came from Javier's lab, where they reported this granularity in terms of the loss of parvalbumin, and then they combined that with ECOG studies that they did perioperatively. And what they did was they classified channels as either spiking or non-spiking, and then took samples from either a non-spiking channel or a spiking channel. And unfortunately, they didn't see a correlation between, for example, the loss of PV and the spiking channels. My feeling is that, because they were using these macro electrodes, the spike could be coming in from anywhere. It could be propagating in from a large distance. And again, this comes back to the very nice poster from the people in Natal that's being presented at this poster, where I think we need to use a much higher resolution type of recording, which allows us to pick out these small regions of the neocortex which are implicated in epileptogenesis. Okay?
I have a question that's related to Mark, which is basically, so are you looking at spikes, and are you looking at synchronization between spikes? Because it might be in the Utah electrode, it might be a really good thing to look at in the red area versus the other area.
So what I should say is the red area, so that zone that I showed where the power map is very hot for the high frequency oscillations, you do tend to see those associated with spikes, but there are spikes that are occurring outside of that as well, but don't have the VFO associated with them. I agree, yeah. And again, that might be the synchronization of those large sharp waves may be simply due to the gap junctions between the interneurons, because it's that long-range connectivity that's provided by those interneuronal networks.
Yeah, if you touch, well, you just mentioned about the characterizing, Rodrigo questions about characterizing very fast oscillations. And I have two questions for you, Mark. The first one is whether have you ever recorded fast oscillations between different channels of the Utah microelectrode? And then, well, I believe you can compute whether they are synchronized or if they have a lag, and then you can somehow trace a pathway between different channels. So can you see, for example, that very fast oscillations are concentrated within the absence of palpable mean positive neurons, and when they move towards the palpable mean region, they start, I don't know, reducing their frequency, or is the glissandi part of that? Maybe like moving very fast oscillations. And my second question is, have you ever tried to stimulate, let's say, layer 6 or layer 1 in order to tackle which are the connections, like intracortical connections, that might be responsible for the generation? So in that sense, do you believe it's possible to produce very fast oscillations in the normal brain using stimulation of different regions?
So your first question, it's an excellent one, and that's, I mean, we've got the data, so we could go back and analyze the data and look for that. I think that's a fantastic suggestion. The second question, we haven't tried to stimulate yet. We can produce very fast oscillations, but you have to really do some pretty nasty things to the slice in order to produce them. And one of the things that you have to do, not surprisingly, is block all of the classical chemical synaptic connections. So if you apply a GABA-A blocker, NMDA blocker, AMPA kainate, GABA-B, and then what you can do is you can micro spritz on CSF, which has got a much more alkaline pH. So we know that these gap junctions are very sensitive to pH changes. So if you spritz on this alkaline ACSF, you then can produce these types of very fast oscillations. And again, we've done that in normal rodent brain slices. They tend to be they're in our human brain slices, as you probably saw from the data, they're quite discrete, the high frequency oscillations, whereas in this with this microspritz of alkaline CSF, the duration tends to be longer. So yeah.
Yes, I think we need maybe another kind of symposium in order to sort of answer some other questions. So my point is not to answer right now, but I think there are at least two big things that were mentioned. It would be nice if you have a quick comment on that, each one of you. Slow events, not exactly fast activities, like slow waves or calcium waves or something which may be not wired, volume transmission, or that discussion on how to synchronize a faraway point or target which is distant from the focus. And in a complex systems view, you can't explain just through synaptic wiring. There is a nice paper, I think, by Fuchs and Agnaci, I think the 80s, talking about that difference between the so-called wired transmission versus volume transmission. And this ultra-slow activity, which is maybe part of the synchronization needed for memory processing and cognition, which is obviously not explained just through synapses.
Yeah, so, in addition to the slower events, the sharp waves, as you've alluded to, there is also very slow activity such as pathological delta, which is known to be implicated in epilepsy. And again, I come back to glia and astrocytes, and we know that, for example, there's a gap junction network of glial cells through which calcium waves can propagate that play a role in these slow waves. And
most likely there's a contribution of the vascular system to that as well. And, you know, these are very important. I think whilst a lot of people have focused, and rightly so, on these very fast oscillations, we also need to think about slower oscillations as well, and build up a much more global picture, because you know, epilepsy is something which propagates over large distances, and in order to understand how seizures evolve, how seizures evolve over longer time scales, for example, that bring in other areas. So there might be an initial lesion in the hippocampus, but we now know that there is this kind of march of the pathological activity, and whilst initially the hippocampus might be the initial site of the lesion, that, you know, as Ed's work demonstrates, you then also get these neocortical foci as well, that are secondary to a primary hippocampal lesion. So I agree that, you know, we need to take a much more holistic approach. Okay, it's now coming up to 11:30. I hope that this has been a very useful discussion. It's been very enjoyable for me. I'd like to take this opportunity to thank both of the speakers for their contributions today, and also thank the audience for their contributions to what was a interesting and illuminating discussion. So thank you very much.
Okay.